Okay, well, thank you for that really, really nice introduction. Yeah, Koramit, Dr. Supapat, if I say it correctly, I have become great friends, and I'm excited so much to be here in Thailand and also to see him almost equally. So, yeah, as was described, I'm really passionate about pediatric brain tumor treatment, and I think the way we're going to progress that is hopefully through somewhat immunotherapy. And so we have somewhat different directions in my lab, but when he invited me here, I said, okay, let me try to pull out what we're doing related to the HLA and immunogenomics. And so it's not Thailand, but I just show you a picture. If you ever come visit Pittsburgh, probably we have nice whitewater rafting, and also the most popular thing perhaps is we put French fries on our sandwich. Try it. So yeah, we do some different things. Like a third of my lab does radionuclide studies, targeted radionuclide therapy to induce immunotherapy response. But then also looking at the T-cell receptors and HLAs across the pediatric brain tumor. And so I have different aims in my talk. I'm first just going to give you a background on pediatric brain tumor because they're very different from adult brain tumor. And you guys have expertise here on that, but some people may not be as aware, and I think it's so important. And then I want to talk about our work looking on T-cell receptors and how we can use that in pediatric brain tumor for looking at patient prognosis. If T cells are there, is that a good thing? Does that help the patient? And then this is like lots of it's unpublished, trying to make some collaboration, but I'm trying to get into the HLA allele space and look at the importance of that for the prognosis and looking at the opposite side of the T cell response. So just to get right into it, so yeah, we have about 700,000 Americans with a brain tumor, and right now the most common in the adult is the IDH wild type glioblastoma, that was referred to earlier today. Survival has not increased much for these patients. Okay, so this is bad. That most of the adult brain tumor is of the malignant one is an IDH wild type glioblastoma. But in kids, again, it's very different. So right now, the brain tumor is the leading cause of the cancer-related death in kids. And so here, this is the red. This red shape is the all malignant cancer. You can see third most common cause of death is cancer in 5 to 9 and 10 to 14-year-old. And then here's the data on the pediatric brain tumor, a lot owing to the improvement of the CAR T for the leukemia. Right now, the pediatric brain tumor is the most common solid tumor and the most common cause of death in kids. And so it's a big, big issue. And we cannot think about it, like I said before, as an adult tumor in a kid. And this should have been obvious, and maybe it wasn't for so long, but children, the tumor actually more commonly is the infratentorial region of the brain. In adult, it's the supratentorial region of the brain, much more common. The types of tumors that occur in kids and adult is different. For example, the ependymoma is a 1.9%, but it's the third most common tumor in kids. The mutation, the genetic drivers of these tumors are also very, very different. In adult, you have this growth factor receptor amplification, very, very common. It's the P53. In the kids, actually, a lot of these are epigenetically regulated tumor, like a histone mutations. Okay, so we have different kinds of tumors, but then within the tumors, and there's so much work being done on the molecular characterization of these tumors that there's just so many of them now, it may even be a little bit hard to keep up with. But it matters, because you can look, this gray box shape over here. Oh, you can't see my thing, but okay. Anyways, you can see just the spine. Oh, okay. You can see for even the posterior fossa ependymoma, if the patient has the type A, then they're probably going to do bad. Similarly, for ependymoma, supratentorial ependymoma, they can look exactly the same. But if they have the Relay fusion, patient's probably going to have much worse outcome than if they have YAP1 fusion. And so the genetics of these tumors are very complex, but also very important. We recently start looking at the immunology between the tumors, and we see that some of these tumors they all have very different immune phenotype. Like, ATRT and DIPG are probably the most two deadly pediatric tumors. These kids may only survive 1 year about. But actually, DIPG has very, very low expression of all the HLA processing machinery, HLA expression, while ATRT, this is one of the highest of all the pediatric tumors. So again, understanding what's going on, I think, is important. I don't need cell immunotherapy to the field, but we're hoping in the kids to get the durable response with immunotherapy. And I think this immunotherapy is one of the few treatment that can hopefully get us there. And then, because I'm going to focus on my talk, this whole idea of what is immunotherapy, if everyone here probably knows, but for the oncolytic viral therapy, for the CAR T, the checkpoint, really, this Hans Bishop guy said really nicely, the immunotherapy, the drug, is the T cell. What you're giving is just trying to boost the T cell for many of them. And then, in case anyone is not aware, the issue with the immune trafficking to the brain is not really an issue of can the cells get in, it's more of what's coming out. It's an afferent versus efferent immune system issue. So T cells can actively migrate into the brain. Antigen getting out of the brain is perhaps a greater issue. Okay, but that being said, if you look at the phase 3 trials for immunotherapy in adult brain tumor, basically everything has failed. Checkpoint hasn't worked, DC vaccine, and there is over my short career so far, I've seen a few times, like, phase 1 trial looked great, phase 2 trial looked great, phase 3 trial, it doesn't work. And so, we need to really figure out how we improve everything so we don't just keep on this cycle. And okay, so now I want to talk about cell therapy because it's one of the topics of this meeting, and Dr. Superpot has really been doing remarkable here with the cell therapy for DIPG. And so I want to share my background with cell therapy. So I mentioned the DIPG tumor. This is diffuse intrinsic pontine glioma, also diffuse midline glioma. They changed the names for no reason or some reason, but this occurs in the kids in school, 5 to 10 years old. And then once they have the tumor, they have 1 year left to live. The radiation that these kids get actually make them better for a little while. It doesn't give them more time to live, but they feel better. That's when they go to Disney World and enjoy the best time. So any intervention during this time has to be very carefully considered with, ruining their few good months they have with their family, and is the treatment going to do anything for them? And so we have to be very careful with what is considered. So, okay, now that DIPG used to be really inoperable, but now they do some biopsy and autopsy studies, and we know that the DIPG tumors have the histone 3.3 mutation called K27M. Okay, so if you have a mutation, this is the potential for a neoantigen and something recognizable by the immune system. And so in this study, we evaluated first, okay, can this, a peptide encompassing this region be immunogenic? And then can we clone the TCR and make a TCR-T? And in short, in this paper, we did that. We did this prediction binding. We took the short peptide, And here you can see with the flow, we can generate tetramer positive cells, and also that these cells can kill in an HLA and antigen-dependent manner. We generated the TCR-T using actually the Takara system, where you use the siRNA to block the endogenous TCR. And then in the mouse model, we can see here, okay, so mice that get the TCR-T, but not mock transduced cells. This is one animal, and here's many. Okay, this works. So yeah, we did this, and then at UCSF they started the trial for the peptide vaccine, and now they started with the HLA-A2 TCR-T vaccine. But there's still many barriers. We're not just trying to treat HLA-A2 positive DIPG. This is a few hundred patients a year. We're really trying to expand way beyond that to 150 tumor types. And if we just go one by one, we will, so many kids, and I think we won't make enough progress. So thinking again about what the T cells can recognize. So, it was mentioned earlier to this, viruses in tumors, and that's probably a great target, highly immunogenic. But this last example I showed you, a neoantigen from a mutation in the tumor. Also, tumor-associated antigens was mentioned in the talk before this. So, okay, so how the T cell recognizes, most people may be familiar with, but is through this VDJ recombination that leads to maybe 1 quadrillion possible T cell receptors. Okay, so this sounds like so many possible T cell receptors. How we start to get a grasp on it. So, real great work from Mark Davis group, Stanford, showed that the similarities in the CDR3 sequence, so this is actually probably one of the more important things to understand for the rest of the data, is this CDR3, if you look over there, that kind of orange, that is the part of the T cell receptor that engages the antigen. And so what Mark Davis's group showed that the CDR3 homology between T cells and TCRs could suggest that those T cells share a same antigen and recognize a shared antigen. And so we decided, we've been collecting with a consortium thousands of pediatric brain tumor patients. And so we used the transcriptomic data, and we pulled out all the CDR3 information from, first we just took 1,000 pediatric brain tumors, now we have 4,000, and we developed a clonal expansion index based on the CDR3. So we look at how many unique CDR3s there are, and then we look at the total predicted T cell abundance in the RNA-seq, and we say clonal expansion index slightly adapted from what's been done before. And then we apply this to the 1,000 pediatric brain tumors initially. And what we found was that, okay, just by looking at the data, this is not mathematical all the way over on the left. But just by looking at the data, okay, DIPG, our worst tumor, we see, okay, has very low clonal expansion index. And some other tumor, like a low-grade glioma, is more towards the other side. NF tumors on the other side. So we say, hey, okay, is this mathematically, does this work? And so we separated the patients by strata, quartiles, and actually we can see the patients with a higher clonal expansion index do much better than the lower. And this is on all the patients. We looked at then each tumor type, low-grade glioma, medulloblastoma, high-grade glioma. We look at them separately and we say, okay, does this hold true? And yeah, you can see blue highest, green, then red, blue, green, red. And when we do this across the tumors, we really see this. So then we looked within a tumor type, within medulloblastoma, we took in collaboration in a collaboration with another group, and basically we can see within the even the types and subtypes of medulloblastoma that this works. So this actually work was recently accepted for the Science Translational Medicine, and they ask us to do tons and tons of Cox pH, logistical like regression models, and so this is just some of them. I'm not going to go into that for the sake of time, but overall we do see, and we want to keep expanding our data set, but to show that the clonal expansion is an important feature associated with the outcome in pediatric brain tumor. So if clonal expansion is important for response, probably those T cells are doing something, right? And so my earliest training was on vaccines for cancer. And so we say, hey, most people take the tumor when they develop the personalized cancer vaccine. They pull off the peptides and they say, hey, can we use this for a vaccine? And some form of that. We said, hey, can we use the T cell and information from the T cell to identify the antigens within the tumor? So I'll walk you through the pipeline in like 30 seconds, hopefully. But what we do is we take all our CDR3s and we group them based on homology across patients. So 2 patients have the same CDR3. Then we group them further based on shared HLAs. We then make a control group of patients that don't have the CDR3, but have the same HLA and same tumor type. And then we say, what are the genetic differences between those patients that have the CDR3 and that don't have the CDR3? And then we say, hey, maybe those are the antigen. And then we apply some binding prediction and machine learning tools that were developed by other groups to try to find the antigen. And so here is actually what the data looks like. So this is just 9 clusters. So you can see cluster 1 is, let's say, the top one. 44 patients have this same CDR3 homology. And there's the tumor distribution. And then what you can see here, I'll just highlight in C2 and C8, for example, you actually see enrichment, statistical enrichment of specific HLAs within that cohort. And then going from there, we predict antigens and the peptides based on comparing the patients, let's say in C2 with a low-grade glioma to other HLA-matched other HLA-matched patients that don't have that CDR3. Okay, then we do wet lab validation. Here we just show that, okay, healthy donor PBMCs, when we stimulate them with the predicted peptides, because these are actually mostly tumor-associated antigens in these pediatric brain tumor, not neoantigens. But we show that, okay, this actually can elicit an immune response. There's not a tolerance, at least in what we're testing. And then more importantly, we isolated the tumor-infiltrating T cells from patients and just stimulated them with a peptide, short stimulation with a peptide, and do ELISpot. And here you can see, okay, when we stimulate the healthy donor PBMC and do ELISpot, as expected, we don't see any response. When we take the patient PBMCs, we see maybe a little bit for some, but not much. But then we can see many spots. The plate looks completely different when we take the TIL and stimulate them with the peptide in some patients. So we can detect our predicted antigen. And so this suggests that the model is working. And additionally, one of the antigen we found is PRAME, which is already a well-established antigen for tumor, which give us further confidence on our approach. So just to summarize this part, I do think we need much more data, but that, in addition to the molecular classification, the histopath classification of tumor, at some point we may have an immunologic classification for tumor, both for prognosis and for suggesting the therapy that should be employed. And also, I think that we can maybe incorporate a T cell in the tumor into our design of peptide vaccine, and then even into the development of TCRT for both personalized and generalized type approach. So I have about 5 more minutes, and so I knew I'd be out of time, so I just kind of summarized this next part. But this is where we get into HLAs. So Zhu Zhen was a visiting scholar in the lab, just went back to China. This is my schematic of a TCR MHC. Not super pretty, but I try to make it kind of simple. But so prior part of the talk focused on TCR. Now is really the HLA peptide MHC. So this is what I think we found. This paper we just revised and now is under review again. It takes so long nowadays try to get things published. But we looked at the HLAs. This is actually an adult and pediatric brain tumor. And what I would say is that we don't find so much generalized things about HLAs in prognosis and incidence of gliomas. But what we do see is somewhat that for prognosis, the capacity to recognize acquired antigens, an acquired mutation-associated epitopes is what's important. And for a benefit for incidence, actually the ability to recognize the early tumor mutation seems potentially critical. And then additionally, we looked a lot at the HLA homozygosity, which is the patient has, if you're not familiar, 2 of the same HLA alleles. So instead of 2 different HLA-A, they will have the same HLA-A, which actually would give you less ability to recognize antigen. And so we look at HLA-As, and we see that the common HLAs in Asian population that's homozygous are more able to recognize driver mutations, and that's associated with a lower incidence. So just to give you the main 4 messages in 3 minutes. Number 1, HLA allele distribution in glioma patients resembles their general population. Okay, up here you can see these are the 3 cohorts. CGGA, this is our largest Chinese cohort. TCGA is a white cohort. CBTN is a pediatric white patient cohort. And you can see here is the more frequent in the glioma patient, and those are the same ones in the normal population. We actually then compared, and what was the adult and pediatric across our cohorts, the HLAs. And the only interesting thing I think for me was that in the kids, we see this HLA-A 1130, and I don't know what it means, but it was actually in like 7% of kids had this, that with brain tumor had this HLA 1130. Okay, so that's message number 1. Yeah, so message number 2, we looked at, okay, can we find any HLAs associated with glioma risk? And yeah, we can. This is, different different statistics, and we compared it to both real world and imputed data sets, and we can find some HLAs associated with glioma risk, but they're not associated at all with immunologic features. So if we take any of these HLAs and say, hey, the ones that are associated with a better risk, well, do we see better predicted high affinity neoantigen? No. And is it more HLA? No. Are there more T cells? No. Is there more immune exhaustion? No. Nothing seemed to be associated with it. So that was message number 2. I think this manuscript has so many figures, and it's even hard to squeeze it onto a slide. But what we find, okay, is that the HLA homozygosity. So if you look between Asian population here and white population here, this is just HLA-A, but you can see the frequent HLA-As that are homozygous here, and it's the most common ones. It's an A201 in the white population, and in the Asian patient, you can see HLA A1101. So if you take all the ones that are homozygous, they have the same ability to recognize predicted antigens in the tumor. And so we say, okay, what could it be? And so